In this video, we will be modeling transcription and translation. If you don't want to model both in one sitting, you can just save your items at the end of transcription for later, and then you can do translation at another time. Here's a list of items you will need to gather to carry out the exercise. The one thing I'll mention in terms of the writing utensil aspect, the pencil, you really do need it because you need to erase at certain times. But in terms of the marker situation, you'll see that I use different colored markers when I do the exercise, but it's not a big deal if you don't have. So to begin, out of the 5 pieces of construction paper you gathered, just take one and fold it long ways, then fold it long ways again, and one more time. Then out of the 8 sections that are there, I want you to cut off just 2 from one end and refold those 2 along the fold line. Next, keeping the folded sheet vertical, I want you to then fold it down from top to bottom, so you're basically making a horizontal crease in the middle. Then you're going to repeat that process, starting at the crease part, fold it down to have it again to form a new crease in the middle. And then you're going to repeat that process one last time, folding from the top of your new crease and folding down, like this would be your 3rd time altogether. So it's going to be actually kind of tough because it's a lot of layers right now. So when you open it up, basically you're going to have 2 sides with 8 sections each. And we're going to write letters on one side for starters. And when I say letters, these letters are bases, so nitrogenous bases. So we're basically going to start with drawing out 1/2 of the DNA. So this whole blue structure is going to represent DNA. So right now, orientation-wise, we are in the nucleus, because that's where DNA is found. And so we're going to take this strand of paper and we're going to create a small-scale DNA model. So starting at the top left and working down to the bottom left, I want you to write out the exact abbreviations for the bases as I'm providing, in the order I'm providing, because that will allow you to follow this entire tutorial and have the same results as me. You also need to make sure there's only 3 letters per section. That's going to be extremely important for later. So each A stands for adenine, C stands for cytosine, G guanine, T thymine. So go ahead and get them all written down. I used a marker, but I mean, if you don't have a marker, anything works. But what we're going to do is after you're done writing down all the letters, I'm going to ask that you pause the video and think to yourself, what are the base pairing rules? Once you've figured them out, you can unpause, and then I tell you it's A goes with T, and vice versa. T goes with A, and C goes with G, or G goes with C, so it's vice versa there as well. So now what I want you to do is pause the video and try to fill out the whole right side following those rules. I want you to try it on your own first, and then when you unpause, you can come back to see the actual answers to check your work. All right, let's check your work. On the right side, you should have A, T, G, C, G, A, C, T, T, G, C, C, T, A, C, A, T, G, A, G, T, G, A. Now I just want to point out that in reality, these bases that we wrote down have hydrogen bonds between them connecting in the middle. And on the outside edges, each base connects to a deoxyribose sugar, since this is DNA, that alternates with a phosphate group repeating, meaning DNA is not just made up of nitrogenous bases, but there's a sugar phosphate backbone. So if we're looking at DNA as a structure that we're kind of drawing kind of like a ladder that then would get twisted around as a double helix, which we're not doing, but that's the way it is in reality. I just want to make sure you guys are remembering and keeping that in the back of your mind. Though obviously to simplify things, we're just going to be looking at the bases only. Next, take a piece of construction paper of a different color and then basically take your DNA structure, fold it in half so you're only seeing 1/2 of that model we just created, and take that and use the other colored piece of construction paper to fold to make a strip that's the same size of just the 1/2 of the DNA structure. Where we're headed with this is we're going to go through transcription, model transcription first, and in transcription, DNA is used as a template to create a strand of messenger RNA. But RNA is single-stranded. It is not double-stranded like DNA, so we only need to create this as a 1/2 size. When you've folded that 1/2 size and you have a good crease, cut at your crease line so you can create the single strip. And just like we had done with our DNA model, I want you to fold that strip down in half once, then another time, and then finally a third time. So when you open it up, just like you had for the DNA, you'll have 8 equal sized sections. Now, if we were only modeling transcription, I would say to simply line up the mRNA single strip next to, like, cover up the 1/2 of the DNA, the right half, and we're going to use the left to use as our template. But the thing is, since we're going to actually also model translation, it's going to be a little bit harder if we write the letters vertical down the way we did for DNA. So I'm actually going to ask that you flip your both DNA and this new colored strip that will be your messenger RNA horizontal. So in this case, what was the left side of the DNA will now be on the bottom, and what was the right side of the DNA, you can't actually see it because it's covered up by this new strip that will be the messenger RNA. Because during transcription, when the mRNA message is like basically being created from DNA, it's only going to be using 1/2 of DNA as the template to do so. But before you start writing the letters to represent the bases on RNA, I want to remind you that the base pair rules are not fully identical in RNA as they are in DNA. So please think to yourself, out of the A to T, T to A, C to G, G to C rule of DNA pairing rules, which of those is different? Because one of those 4 letters is not present in DNA, but instead a different letter is present in RNA. So pause the video and then come back so we can review it. Okay, so what is not in RNA is T, thymine. Instead of T or thymine, RNA molecules have U or uracil. So when you go to write out this code, and I'm going to ask that you write it out horizontally from left to right, but again, only 3 letters per section that correlate with the DNA that you are provided with. But what happens is, if you see an A in DNA, you're going to write a U in RNA because there's no T in RNA. If you see a T in DNA, you might think, well, what do I do there? It's okay. RNA can make A's, right? And A is what goes to T. So even though RNA doesn't have a T on it, when you see a T, you simply just write A. The nice part about it is the C to G, G to C rule is the same. So if you see a C, you write a G. If you see a G, you write a C. The one thing I will warn you though, and ask that you be careful of, since it's a little tricky, since we wrote out the DNA bases vertically, but then we flipped it horizontally, just be careful because you can get tripped up because the C's in DNA, when flipped this way, might look like U's, but remember, it's not a U, it's a C. C goes to G, so if you see that, you just write a G when you're writing out your RNA. And the other thing to remember is DNA doesn't have U's, so you know you're not looking at a U. I just mentioned it because as I started writing it out, I'm like, oh, I caught myself doing that. So I'll help you here with the first one. We have TAC on DNA. So based on the rules we just reviewed, that means we need to write horizontally AUG in that section. So now I want you to try the rest of the sections in sets of 3 by yourself. So pause the video and then come back so we can go over the answers. All right, let's start going through them and seeing how you did. course, you started with AUG, we gave you that one, but then you should have had CGA, CUU, GCC, UAC, AAU, GAG, and UGA. So we have successfully modeled transcription. The orange piece, the messenger RNA, will now leave the nucleus, head into the cytoplasm, and try to find a ribosome because ribosomes are the location where protein synthesis occurs. And that's where we'll pick up with translation. But before we do so, I want to just make sure everybody is reminded that just like DNA is more detailed in structure than just bases, RNA is obviously the same situation. RNA, it's single-stranded, yes, which is different than DNA. But when we talk about a sugar phosphate backbone of DNA, RNA actually has one as well. Now, of course, it's only a one-sided thing, so it's only on that one side. But there's another key difference. Instead of the sugar being deoxyribose that rotates with phosphate groups, in RNA, the sugar is ribose. So you see R here for ribose. If we were to make a more detailed model, it would be something more like this, where each base connects to one of those ribose sugars, but then between each sugar is a phosphate group. But this is, of course, where RNA gets its name, ribonucleic acid, because it contains the sugar ribose, compared to DNA's deoxyribonucleic acid, because it contains the sugar deoxyribose. But again, we'll be keeping things simple, so we'll just use the bases on both RNA and DNA to carry out our simulation. If you are not planning on doing the translation part of the simulation right now, you can put your supplies away in a safe spot to come back to the video later to do translation. We have completely modeled transcription, and at this point, we've got a transcript. We've got code in the form of a message in messenger RNA that needs to get decoded so a protein can be made during translation. So to begin translation, I want you to place that messenger RNA code onto a different colored sheet of construction paper. When the messenger RNA leaves the nucleus, it's going to try to go to a ribosome. So think something like all those ribosomes embedded on the rough yar. There's also ribosomes throughout the cytoplasm as well, but it's going to go and find a ribosome. So what we need to do is we need to draw that the messenger RNA has reached a ribosome. So what I'd like you to do is I want you to move your transcript, the strip of mRNA code, and I want you to shift it so the first three letters, the AUG, are kind of towards the middle of the page, but then it's basically spread out to draw what looks basically like a hamburger bun, meaning ribosomes are not perfectly spherical shaped. I know when we draw them as little circles when drawing the cell, but the reality is they have a large subunit and a small subunit. So I want you to draw what looks basically like a hamburger bun, but make it so the hamburger bun would fit 3 sections of those 3 pieces of code. So if you can see here, you're not going to write 1, 2, 3. I just wanted to put that on the picture so you understand. So what I'd use my green marker to draw the top section and the lower section, that's all I want you to draw. Then label it with the actual word ribosome. So during the whole modeling exercise, you remember that that is the organelle that it's representing. Next, take a piece of construction paper of a different color, fold it down in half, and with the uncreased side, I want you to place that right on your model of the ribosome with messenger RNA. Place it right on top of the last 2 sections, like align the paper, meaning of the 3 sections that are actually shown on the ribosome. Slide it over so it's right above the last 2. And then the third one of those last 2, I want you to basically look at the amount of space that those 3 letters take up, meaning, in other words, it's the space of 1 section, and put 2 lines. So then that's telling you how big you want what you're going to be creating next to be in terms of how far across. We're going to be making transfer RNAs now. So you're going to take besides putting 2 lines, and you can see here they're vertical lines as to what will fit a space of 3, you're going to take your marker or whatever you're using and you're going to make a horizontal line towards the top of the ribosome to show how tall you want the tRNA to be. And it's not really a big deal. It's just if we do them around this size, the bottom part, the 3 does matter, but in terms of the height, that's not really the end of the world if it's not perfect. But do the best you can, because then what we're going to do is we're going to create basically a bubble letter T or a cross, however you want to describe that, and you're going to cut it out to get 2 different things that look like bubble-shaped T's or crosses. Since the marker outline will only show up on the one that was on top, I just like to outline the other one as well. I mean, you don't have to. So then I outline both. Now we're going to label both of those T structures tRNA, so lowercase t and a capital RNA to stand for transfer RNA. So the thing with ribosomes is they don't make proteins by themselves. They need help. They need help from transfer RNAs. Transfer RNAs are going to be a delivery system to bring in amino acids, the building blocks of proteins, to make proteins. There's going to be a whole bunch of different tRNAs bringing in different amino acids. We're going to creatively just use 2, and this is why we wanted a pencil with an eraser. So we want to put different code at the bottom of it, and I'll explain more about code in a little bit, that we can erase it and change it to make certain tRNAs hold certain amino acids. So now what I want you to do is place one of the tRNAs on top of the first 3 letters of the messenger RNA. And if you notice, I put them at the far end of the ribosome as if the ribosome was moving from left to right when it's reading that first section of the messenger RNA. And the reason I put a little question mark at the bottom of the tRNA is because it's going to know to come in and bring in a certain amino acid based on whatever code is on the messenger RNA. So when I say based on the code in messenger RNA, you see this first segment says AUG. So I want you to think to yourself, what would be at the bottom of this particular tRNA that would match up with base pair rules for RNA in terms of what letters, what 3 bases would be at the bottom of this tRNA? Think about it, pause the video, and then come back. If you said UAC, you're absolutely right. And that bit of code at the bottom of a tRNA molecule is called an anticodon. And it correlates with the code, that section on the mRNA and those 3 letters pieces. So it reads based on, like the ribosome reads the mRNA based on sections of 3 bases. On mRNA, it's called the codon, so that's the code that's getting decoded. On tRNA, it's called the anticodon. Now, based on which anticodon and codon come together, the particular transfer RNA will then bring in a particular amino acid. So, and before I teach you how to use a codon chart to figure out which amino acid per which tRNA, we need to actually make some amino acids. So take a piece of construction paper of a different color, fold it in half long ways, and after you've creased it in half that way, then I want you to fold it down from top down. And then when you fold it down, top down, then I want you to fold it across. So basically we're going to fold it 3 different times, because ultimately when we take a circular object, a small circular object such as a quarter works really well, and take a writing utensil, outline that quarter or whatever you're using, maybe like the bottom of a plastic bottle or something. You can freehand it too if you don't have something to trace it, but you're going to end up cutting out, and based on all the layers from the folding, 8 different circles which will represent your amino acids. Granted, you're only going to need 7 for this exercise, but there you go, you get a little bonus circle. All right, so now we have to talk about figuring out what should be written on the circle for our first transfer RNAs, like which amino acid is this first transfer RNA, bringing in based on the codon that was decoded by the anticodon on the tRNA. So when I specifically reference codon, it's because we're going to be using a codon chart to figure out the amino acid, not an anticodon chart. There is no such thing. We use codons on messenger RNA. So it gets a little tricky because both RNA types, because transfer RNA is RNA as well, use A's, use C's and G's, which you could use on a chart to figure out and decode for a protein, but if you use it, you'll be wrong. So you need to make sure whatever three letters you're looking at to figure out which amino acid is associated will be the letters on messenger RNA. So our first set of letters was AUG. We're going to teach you in this video how to use both a circular as well as a rectangular codon chart. We're going to start with the circular chart. I just pulled both of them off of Wikipedia. So the way the circular codon chart works is it's basically you begin in the middle of the actual chart and you work your way out. So our first letter was an A for adenine, so we start with that base in the middle. Then we work our way out, so we find our U in the next layer, and then we keep working our way out to find the G in the final layer. And then we keep looking past and we see that it's associated with methionine or MET. So this particular chart is using abbreviations for amino acids, which is great because when we want to write out the names of the amino acids on the circles, abbreviations will be a lot easier. So let's do it. Take a circle that's representing an amino acid, place the circle on top of the tRNA because that's what's going to be carrying it, and write MET on it. In that case, because this is methionine. And tape it on there. Methionine is always actually the first amino acid, though it may or may not be removed in a given protein, and that's because AUG is considered the start codon. You may or may not have noticed, but when we looked at that circular codon chart, there was a symbol, and that triangular symbol represented the start codon. Now that the first amino acid is present, imagine the ribosome moving from left to right, so it's going to slide down where now AUG is in the middle position because a new transfer RNA is coming in to coordinate with the second codon, the CGA. So I want you to think to yourself, what would the anticodon be? Pause the video and then come back to check. If you said GCU, you're absolutely right. But remember, we don't look at the anticodon to figure out the amino acid, we look at the codon. So we need to look at CGA. However, this time we're going to take a look at a rectangular codon chart. Where you start is on the left side. That's where you look at the first base. So in this case, it's the base C. Then you have figured out the row where you're at, but you need to next figure out the column. So to do this, we look for our second base along the top, in this case G, so it's the last column on the far right. So when we work our way down, we have it narrowed down to the general location, but we have one last base we need to look at. So for the last base, we look along the far right, and then we just go across to that box where we had figured out the row and the column, and in this case, it links to arginine or ARG. And you might be thinking to yourself, well, couldn't I just have looked on the entire chart until I found CGA, and then I would have figured out it linked to arginine? In this particular case, that's technically true because this particular codon chart, that's the rectangular version, does give you every three-letter combination. Now, obviously, it'll take you a lot longer than using the left on the first, the top across for the second, and then the far right for the base. For the third base to figure out which one. But, the thing you have to be careful of is some codon charts that are rectangular, they do not actually give you all the codons listed out, and you have to only use the left, top, and right letters to figure out which amino acid. The other thing you have to be careful of is sometimes certain amino acids, like in this one, you could tell that there is multiple code to get to arginine. So even if you just figured out the first two letters, technically, without even looking for the third letter, you would have figured it out. And although that is true in this particular case, you also have to be careful because it's not always true. Just because the first two bases are the same does not mean it'll code for the same amino acids. You really have to look at all three bases to determine it. So go ahead and take one of the other circles for amino acids, write ARG on it, tape it onto that transfer RNA, and then visualize the ribosome working forward. Keep going down from left to right. So at this point, the two transfer RNAs are now they're kind of shifted down too, if you kind of visualize how this goes, because ultimately what we want to show is that ending one, that had the MET on it, it's going to transfer, like MET's going to leave that first transfer RNA. It's going to connect to ARG. The transfer RNA that was carrying ARG is now going to carry both amino acids. The other transfer RNA, because it's not needed anymore, is going to leave. But because we only created 2 transfer RNA molecules, we're going to keep repeating this process to decode our whole messenger RNA, but we're going to basically essentially continuously kick out the end, like the farthest exit point, that particular transfer RNA. We're going to erase the anticodon that's present on that one. We're going to figure out what the next one's anticodon is, but then of course we're going to use the codons to figure out the amino acids. We're going to repeat this until we get to the end of our messenger RNA transcript. So keeping that one transfer RNA that you had attached the MET to the ARG, like you just transferred the tape, hopefully you figured that out, to just move it and use the same tape you had. But then you're going to put that one, the new transfer RNA, we're saying new because we erased it so we can reuse it, but we're going to bring that one in to the right position, meaning if we have 3 positions like kind of carved out on a ribosome the way we set this up, then I want you to think to yourself, if the next codon is CUU, what is this anticodon? So pause the video a moment. If you said GAA, you're absolutely right. But keep in mind that's the anticodon, which is on transfer RNA. So now we have to look at the codon, which is on mRNA. So the codon in this case is CUU. So we're going to have to pull up our codon chart to figure out what CUU codes for. So I'm going to give you a circular chart this time. Go ahead and pause the video, try to solve it yourself, and then come back. Hopefully following from int out, you found that CUU codes for Lu or leucine. So write Lu on a circle, tape it to that tRNA, and also move those tRNAs over to the left 2 positions. Then move the MET and ARG, which were on the other transfer RNA, move them over and tape them to Lu. So basically you're getting a growing polypeptide chain. And I'm saying polypeptide chain because each of these amino acids, which in my case I'm representing with the red circles, when they are connected together, they're connected by a peptide bond. And then when you put a whole bunch of these amino acids together by peptide bonds, we call that a polypeptide. But of course, that's the protein we are building. So go ahead and get rid of that transfer RNA that used to be carrying the polypeptide chain. Erase the anticodon that was on it, and take it and put it over to the next codon so we can figure out the anticodon. So go ahead and pause the video to think about what the anticodon is and then restart it to see if you're right. Hopefully you correctly identified it as CGG, which of course correlates with the codon GCC, which you have to use now on the codon chart. So please go ahead and try to pause the video, give yourself some time to figure out the correct amino acid associated with the GCC, and then unpause when you're ready. I'm assuming you figured out it was alanine or ALA. So now write ALA on a circle, tape that circle representing the amino acid on top of that transfer RNA, and then shift the 2 RNAs in general over to the left 2 positions on the ribosome, because ultimately the ribosome keeps moving left to right. And you also need to transfer all that growing chain of amino acids over onto ALA. So now it should read MET, ARG, Lu, ALA, being held on that newest transfer RNA that just brought in ALA. Kick out that unneeded tRNA, erase the anticodon, move the tRNA back as if it's a new one coming in to the next codon, figure out the anticodon, pause the video, come back to check if you're right. I'm assuming you're getting a hang of this. You probably determined that it was AUG, which correlates with that UAC, but of course now you have to use your codon chart to figure out what UAC decodes for. Please pause the video, use the chart, figure it out, and then restart the video to check your answer. I believe in you. I'm sure you got UAC coding for tyrosine or tyre. So go ahead and put tyre, TYR, on another circle, taping it on top of the tRNA. Remember with all of these to keep shifting as if the ribosome is moving down. So now the transfer RNAs will be in the left 2 positions instead of the right 2 positions. Move the growing polypeptide chain over to tire. Kick out that transfer RNA that's no longer needed. Go ahead and erase the anticodon on there. Bring the tRNA back over to the next codon. Figure out the anticodon. Pause the video. Restart the video to check your work. I'm sure you got UUA, which we know correlates with the codon AAU. So now check out this codon chart, figure out what it codes for, pause the video while you're doing so, and then restart the video to check your work. I know you got asparagine since you're a pro now, so go ahead and write ASN on your next circle, tape it to that next transfer RNA, shift those transfer RNAs down to their new position, transfer that whole long growing chain of amino acids over to ASN, so that new transfer RNA has the whole growing chain, kick out that one that's no longer needed. As usual, erase that anticodon since we're going to reuse that transfer RNA. Put that transfer RNA over into the next position. Then pause the video as you are solving for the anticodon, repressing play when you're ready to see if your work was right. And of course, I'm sure it was, since you're fabulous. You would have gotten CUC, but make sure you are using the codon, not the anticodon. So we're looking at GAG on the codon chart. Try it out. Pause the video while you are trying. Restart it up to check your work. Since you get glutamic acid or glue, then you know the drill. Go ahead and write glue on top of a circle, tape it to that transfer RNA, shift the transfer RNAs into the left position as the ribosome is working down the transcriptive messenger RNA, and then affix the growing chain of amino acids to the newest arrival, the glue. Remove the now empty tRNA, erase its anticodon so we can reuse that piece of paper to model this as the next tRNA comes in. And look, we're on the last one on the transcript, so let's see what it says. Go ahead and pause the video as you first figure out the anticodon. When you get that ACU, that correlates, of course, with the codon UGA, so you get to use your codon chart for the final time in this activity. Here's your codon chart. Just pause the video, figure it out, and then come back. Well, now looky here. You figured out that it codes to stop. So there's actually no amino acid when you reach the end of reading that whole messenger RNA transcript, because that's representing basically the end of whatever gene was being read. And so we have completed our entire amino acid chain. So at this point, everything basically just disassembles. The messenger RNA transcript, it's not going to be used again. It'll basically just get broken down, and the different nucleotides that were in it can be, used in the body again or whatever, but it's degraded. But the ribosome, I mean, that's an organelle. It's going to be used again and again to make proteins. The transfer RNAs, I mean, obviously they'll have to get a new amino acid attached to them to bring in, but they will be used over and over again. But of course, our main focus will be this chain of amino acids. What happens with the polypeptide chain? Well, the polypeptide chain will fold up into a unique 3D shape, and that shape will determine that particular protein's function. So proteins have that specialized shape to perform its duties, and we have now completely modeled translation, going from a messenger RNA transcript and translating that into a chain of amino acids that will be a specific protein. Hope you found this helpful. Construction paper biology. Like, share, subscribe, oh, won't you please? Thanks!